Welcome to Let's Learn Plants. Today, we're jumping right into one of the most elegant processes in all of biology. We are going to explore the fascinating biochemical journey of how a simple linear amino acid sequence transforms into a complex, three-dimensional, highly functional protein. Understanding this process literally unlocks the secrets to how life operates at a molecular level. In this explainer, we'll be covering 1. The rules of folding, 2. The folding paradox, 3. Predicting protein structures, 4. Disulfide bridges and stability, and finally, 5. Misfolding and disease. Let's get right to it.
Section 1. The rules of folding. Building the biochemical foundation.
Now, to really understand folding, we first need to understand the destination. The ultimate goal for any folding protein is to reach its native structure. You can think of this as the protein's happy place. It's the lowest energy, most thermodynamically stable shape it can possibly achieve, where all the molecules are just perfectly relaxed. And the fundamental question of biochemistry is this: how does the linear sequence know exactly how to find this one perfect 3D shape?
Well, it starts locally. Certain amino acid residues actually have natural personalities, meaning they promote different structural geometries. For instance, residues like alanine, glutamate, and leucine, they strongly prefer to form coiled alpha helix structures. But then you have residues with branching at the beta carbon, like valine and isoleucine. These actually destabilize those coils because of steric clashes. Basically, their bulky sides just physically bump into each other. So instead, they perfectly accommodate the flatter, extended geometry of a beta strand. But predicting the final structure requires looking way beyond just these local preferences. The context from the rest of the protein is absolutely crucial.
And that context leads us to how the whole structure comes together. Folding is not some slow, piecemeal, one-step-at-a-time process. It's a highly cooperative, all-or-none transition. Because the stability of one part depends entirely on its interactions with the rest of the protein, if you disrupt just one region, you lose those stabilizing forces, and the entire protein rapidly unravels, kind of like a house of cards. It's a sharp, dramatic transition, meaning the intermediate structures are incredibly fast and transient.
Moving on to Section 2, the folding paradox, nature's millisecond miracle.
This brings us to a mind-bending mathematical problem called Levinthal's paradox. It points out that a polypeptide chain has an astronomically large number of possible conformations. To put that into perspective, if a tiny 100-residue protein tried every possible shape randomly just to find its native structure, it would take a staggering 1.6 times 10 to the 27th years to fold. Yet, as we know, small proteins in nature fold in mere milliseconds. No way are they sampling every conformation. They've got to be taking a shortcut.
The brilliant solution to this paradox is a defined pathway driven by something called cumulative selection. Imagine a giant energy funnel. Up at the wide top rim, the protein is unfolded. It's got high free energy and endless chaotic shapes. But as local regions rapidly form correct, stable structures, those specific pieces are retained. They don't unfold again. These partly correct intermediates guide the rest of the protein, rapidly narrowing the options and funneling the whole structure down to the bottom, that unique, low-energy native state.
Section 3. Predicting protein structures, decoding the sequence.
Now that we know how nature does it, let's look at how we decode these structures computationally. There are 3 main approaches. First is homology modeling, which requires a high sequence identity, over 30% identical to a known protein. This gives you the highest accuracy at the lowest computational cost. Then we have protein threading, which is used when your sequence identity dips into the twilight zone below 20%. This relies on the fact that nature only uses a limited number of structural folds. And finally, ab initio modeling. This uses pure physics and chemical principles to build novel folds totally from scratch without any template at all, which makes it computationally very expensive. For those of you utilizing homology modeling, there are 5 vital steps to your game plan. First, you recognize a template in the protein databank using a tool like BLAST. Second, you carefully align your target sequence to that template. Third, you construct the backbone model. Fourth, you predict the specific sidechain angles. And finally, step 5, you refine and validate the model's energy and stereochemistry, basically ensuring the 3D spatial arrangement of the atoms is physically possible and chemically sound.
But we absolutely have to highlight the elephant in the room: modern artificial intelligence. Driven by the biennial CASP competition, deep learning tools like AlphaFold 2 have been a real game changer. They've effectively solved major parts of the template-free prediction problem. By training neural networks on massive datasets, AlphaFold can accurately predict interresidue distances and contacts without needing a template at all. It achieves near experimental accuracy and has literally transformed structural biology overnight.
Section 4. Disulfide bridges and stability. Locking it in place. Okay, so we've got our folded protein, but how does it stay that way? Unlike weak, non-covalent hydrogen bonds, disulfide bridges are permanent covalent crosslinks. They form through a chemical reaction between two nearby cysteine amino acids. You can think of them as the heavy-duty structural anchors, or zip ties, of biochemistry. They provide massive stability and lock that active conformation in place against heat and chemical denaturation. It's really quite amazing. If you treat a protein with chemicals that sever these disulfide bonds, it denatures. It collapses into an inactive, random coil. But here is the wild part. If you remove those chemicals and allow the protein to slowly reoxidize, it spontaneously refolds right back into its original active native conformation, complete with the correct disulfide pairings. This proves a fundamental law of biology. The instructions for the active 3D structure are inherently written right into the primary amino acid sequence. The protein already knows what to do. Now, even after folding is completely finished, proteins often undergo post-translational modifications, or PTMs, to become fully functional. Think of these as the crucial finishing touches. Phosphorylation acts as a reversible on-off switch for cell signaling. Hydroxylation stabilizes structures like collagen, which, fun fact, requires vitamin C to happen. Glycosylation adds carbohydrate units to aid in signaling, and cleavage actually trims the polypeptide chain to activate certain digestive enzymes or hormones.
Finally, Section 5, misfolding and disease. What happens when the process fails?
When protein folding goes wrong, it leads to a class of severe diseases known as amyloidoses. In these conditions, proteins that are normally totally soluble and functional misfold. They convert into insoluble, toxic aggregates that are heavily rich in beta sheets. And what makes this so devastating is that these misfolded proteins actually act as infectious seeds. They physically clump together into toxic plaques and literally force healthy surrounding proteins to adopt the exact same incorrect aggregated shape. The bad shape spreads like wildfire. This specific misfolding process is exactly what drives certain infectious neurological diseases. Take prion conversion, for example. A normal prion protein found naturally in our bodies is rich in coiled alpha helices, and it's completely harmless. But it can dangerously shapeshift into an infectious agent rich in flat beta strands. This misfolded version aggregates into rigid fibers and causes fatal, transmissible conditions like mad cow disease. But misfolding isn't just about infectious diseases. It's also the direct culprit behind devastating non-infectious conditions that impact millions of people worldwide. This prominently includes Alzheimer's disease, which is characterized by amyloid plaques made up of the misfolded A-beta polypeptide. It also includes Parkinson's disease. Current research suggests the primary mechanism of toxicity here comes from small clumps or oligomers of these misfolded proteins directly punching holes in and damaging our cellular membranes. Before we wrap up, we have to acknowledge that biology absolutely loves exceptions. While we spent this whole time talking about the one sequence, one structure rule, some proteins intentionally remain unstructured. These are called intrinsically unstructured proteins, and they only adopt a firm 3D shape when binding to a specific partner. Others, known as metamorphic proteins, exist in an equilibrium, actively flipping between two entirely distinct functional shapes to perform complex signaling tasks. They're basically biological Swiss Army knives. So I want to leave you with this provocative thought. We've seen just how elegant nature's folding process is, and we've seen the devastating diseases that occur when it fails. But as our computational mastery over protein folding grows with deep learning tools like AlphaFold, how close are we to artificially reversing the misfolding that drives Alzheimer's and prion diseases? Could the very same AI that predicts the normal structure hold the key to finally curing the world's most complex neurodegenerative diseases? Keep that question in mind as you continue your journey into biochemistry. Thank you so much for joining me on this explainer, and we'll see you next time.